Okay, so you should be able to see my screen now, right? Okay, then I will start ahead. Like Tina mentioned, I want to start off giving you a little bit of an educational teaser or talking a little bit about the underlying metabolomics data acquisition types and also the approaches. The stuff I'm talking about right now is also applicable, of course, to proteomics, but I want to be focused a little bit more on the metabolomics field, since this is like my main expertise. Okay, so generally we can distinguish between two approaches. One of them we call untargeted or non-targeted approach, and the other one is a targeted approach. In the non-targeted, untargeted, or even called sometimes shotgun approach, this is based on a data-dependent acquisition or data-independent acquisition. Today I want to focus more on the DDA acquisition. And the question that is asked in an untargeted approach is, what is the metabolic difference, or what is the overall metabolic fingerprint of a sample? In a targeted approach, this is more based on targeted data acquisition. And here the aim is to answer the question, what is the quantity of a specific metabolite within a biological sample? So I want to start off with the untargeted approach and go into a little bit of detail on the workflow. This is also something we commonly perform here at the BayBioMS. And the question, like I said, is what is the global metabolic profile of a sample, and how do we get there? We start off with our samples. So commonly used samples in metabolomics analysis are tissues, cells, blood, or other biofluids. And they are, of course, the metabolites have to be extracted, and then they are introduced into our LC-MS system. And what we get out of it is a very overlaid or very complex chromatograms, ion chromatograms, where all the information of all metabolites are contained. And then data alignment and statistical analysis is the follow-up steps, where with the help of statistical analysis, we get this global metabolic profile of biological samples, which can then be used to be compared between different cohorts or between different patients, and in order to elucidate the differences between the metabolome. To keep in mind, so the untargeted metabolomics approach aims to get a comprehensive overview of the entire metabolome. No real prior knowledge about your samples or which analytes you want to analyze is needed, but I want to also focus that there is no real 100% untargeted metabolomics possible. So there's also always a different analytical systems or the extraction methods that will favor or disfavor certain metabolite classes. So please keep in mind, we cannot really depict 100% of the metabolome completely unbiased. So this is always the analytical strategy has always be adapted for the specific questions. Of course, like I mentioned, a thorough statistical analysis is absolutely essential. And in the end, the goal of an untargeted approach often is the discovery of a biomarker, which is significant for the intervention or for a treatment, or can also be used in diagnosis. If we take a look at the actual MS data acquisition in the so-called DDA mode or data-dependent acquisition, first of all, a full MS1 scan or a full scan of the MS1 space is performed. In metabolomics, the mass analyzer commonly used is a time-of-flight system. In proteomics, there's also a possibility using an Orbitrap, or Orbitrap is commonly used. Based on this MS1 scan, based on this MS1 scan, several of the most intense precursor ions are then selected, and from each of these precursor ions, a full MS2 scan is performed, and again, the MS2 spectra is acquired. This is performed for several of the most intense precursor ions. And in the end, we get for each metabolite, we get a MS1 and an MS2. And these informations can then be further used to identify the metabolite, either by scanning or searching through databases, or measuring reference compounds and see if they have matching MS1 and MS2 spectra. The second approach I want to quickly talk about today is the targeted approach. Like I said here, the question often is what are the levels of specific metabolites in question, and talking a little bit about the workflow. Here we start off with the actual standard metabolites, or the metabolites that we want to analyze, that we want to quantify. So at first you need to know what exactly are you looking for, what do you want to quantify, and then you need the actual reference compounds. And first they are infused in the MS system. And then the first part of the targeted metabolomics workflow is the optimization of the MS parameters as well as the LC parameters in order to get a sufficient and robust method. If this first part of the workflow is completed, then you can also prepare the standard curves are prepared, which are used for the quantitation, and then the samples are introduced. Here again, typical biological samples can be used. And after workup step, they are then introduced into the LC-MS system, and they are analyzed with the parameters that have been optimized in the first part of the workflow. And then at the end, the output is a quantity of a specific metabolite within the biological sample. Here to keep in mind, of course, we are talking about an absolute quantification of the metabolites. Here you absolutely need the prior knowledge. You need to know what you want to quantify, and you need the standard reference substances. And then with the quantitative results, you can compare the metabolites within different conditions or over a time course, for example. And as you will see in the second part of my talk today, this can also be used as a follow-up experiment for untargeted metabolomics to verify the findings of the first approach. So before I jump over to the second part, I want to quickly talk about the data acquisition of the targeted metabolomics approach. Here, the analysis is commonly performed on a triple quadrupole mass spectrometer. And so from the compounds, we know the standard metabolites we want to quantify, the typical precursor ion, the M/Z value. Only one M/Z value is stabilized in the Q1 in the first quadrupole and is able to traverse the first quadrupole. This one metabolite is then fragmented in the second quadrupole or in the fragmentation cell. And one of these fragments is then stabilized in a third quadrupole and can be detected in the detector. So what we are analyzing here in the targeted approach is always a pair of Q1 and Q3 masses. This is also commonly referred to as mass transition. And in the end, we get our peak. So in order to add a certain level of robustness or to make the method more specific, we not only use one of these mass transitions, but commonly we use two or at least two. And this is then called the MRM, or multiple reaction monitoring. And this is basically just in a second scan, not only this one fragment is this one mass transition is stabilized, but another mass transition is stabilized in Q3, and we get two peaks. One of them, the most intense, is called quantifier, which is used then for the quantitative determination of the compounds. And usually the second most intense is called qualifier. And this is used to verify that this is in fact the compound we are looking for, because in a triple quadrupole mass spectrometer, we are not having an exact mass, no high resolution mass is acquired. Okay, this is already the end of the first educational teaser. I want to then directly switch over to my talk and showing you an application of both untargeted as well as targeted metabolomics or data acquisition modes. As Tina mentioned in the beginning, I am a food chemist by training. So we sort of borrowed the metabolomics approach and applied them to our food systems, in my case, coffee. So we have no elaborate cohort or elaborate intervention study where we can analyze biological samples, but we analyzed foods by means of the same strategy, basically. And what I want to talk about today is the identification and quantitation of reaction products from quinic acid, lactone, and chlorogenic acid with Strecker aldehydes in roasted coffee. Just very quickly in the beginning, I want to talk about coffee. So coffee is grown in large portions in the world within the so-called coffee belt, or also called bean belt, where the equator is right in the middle. There's about 120 species of coffee, which belongs to the family of Rubicaceae, but only 2 are commercially grown, which is, all of you will know this, it's the coffee Arabica, which accounts for about 60 to 80% of the total production, and the coffee Canifora with the variation Robusta, which is the other 20 to 40% of the world production. In 2020, over 10 million tons of coffee were produced, and just as a bit of an example, the per capita consumption in Germany amounts to 166 liters per person per day. It's about 4 cups per person per day. So you can see the relevance coffee has on our everyday life. Not just in Germany, but worldwide, coffee consumption is on the rise since several decades. And I just last week I read new data, and it is projected to grow even another 1.8% in the next year. One of the major reasons for these increasing coffee consumption is, of course, the very attractive and alluring aroma of the olfactory sensation of coffee. And the actual molecules responsible for this olfactory sensation, they have already been elucidated by means of the so-called synsomics approach. And here at the bottom, you can see 25 so-called key coffee odorants. And when you recombine them in the right concentration, this is what we commonly refer to as flavor reconstitution or flavor re-engineering, then you actually get the aroma impression of a freshly prepared coffee beverage. And while this works very well for, like I said, a freshly prepared coffee beverage, there is one major problem that coffee has, and this is that the alluring aroma that many of us love is really unstable, and within minutes after you prepare the brew, this deteriorates. First analytical studies who focused on this decline of coffee aroma were performed by Professor Hofmann et al. Back in 2002, and he could show by means of GC-MS experiments that the headspace concentration of 2 coffee thiols, the thiols are responsible for the roasty notes of coffee, that they decline significantly when you keep a coffee beverage warm at 80 degrees Celsius. For example, the concentration of furfuryl thiol, which is really the major player of the key coffee odorant for the roastiness, is declined to almost 1/3 within 1 hour. In our working group, many other studies were performed with the aim of elucidating the molecular underlying features or the molecular understanding of this so-called aroma staling, and they were able to account these reactions taking place to either high molecular weight or to low molecular weight compounds found within the coffee brew. Today, I want to focus mainly on the low molecular weight fraction of coffee, but until now, there's still no clear explanation what happens to the aroma or what happens to the entirety of the aroma. And this is actually a big challenge for not only for research, but also for the coffee producing industry, because they are losing basically their aroma during production already. So, and this led us to the application of this untargeted metabolomics approach, because we wanted to get a real comprehensive overview, what is happening to the aroma, where is the aroma reacting, or where are we losing this coffee aroma. And so we used, like I mentioned earlier, this untargeted approach, but instead of having a treatment group or a reference or a control group, we now have a coffee beverage, and one of these beverages or the coffee beverage was split, and the one portion was enriched with Strecker aldehydes and one was kept as is and used as the control group. Both of them were analyzed by UPLC TOF-MS, and of course the statistical analysis was performed to elucidate certain marker compounds. Afterwards, the structure of these marker compounds were elucidated using NMR experiments, and then a targeted approach was followed up in order to really quantify these compounds within the coffee beverage. So to get to the actual targeted approach, here you can see the principal component analysis of 3 different coffee beverages that were treated either with acetaldehyde 500 parts per million and stored for 4 hours at 70 degrees Celsius, typical conditions where you would store your coffee in, if you would store your coffee in a thermos flask. The second coffee beverage was incubated at the same conditions, but no aldehyde was added, and we also measured a freshly prepared coffee beverage. And you can see clearly that the principal component analysis shows that the groups are clearly differentiated, and this gave us a first glimpse that there might be some differences within these groups. And in order to pinpoint these differences between the sample groups, the stored sample with and without acetaldehyde were analyzed or were visualized within this similarity plot. And within this similarity plot, each of the black squares represents a feature of the TOF-MS analysis. And if the feature exhibits high X and Y axis values, this means that this contributes significantly to the overall difference between treated and non-treated, between coffee with acetaldehyde and coffee without acetaldehyde addition. And we then took a look at several of the marker compounds, which are located over here. And I want to focus today mainly on this one marker compound marked in blue with an accurate mass of 217 m/Z. So our next question, or our next step, was we wanted to verify if within this marker compound there was actually an acetaldehyde incorporated. And in order to make this assumption, or to verify this assumption, we repeated these TOF-MS analysis, but we now used deuterated acetaldehyde instead of the non-deuterated acetaldehyde. And what this should cause, if in our molecule there's an acetaldehyde incorporated, this would also cause a mass shift of +3 Dalton, so from 217 to 220. And you can see here on the right-hand side a normalized trend plot where you can see the abundance of the five different UPLC TOF-MS acquisitions. And here is on the top side, we only monitored the intensity of the ion with 217 m/Z in easy negative mode, and on the bottom we monitored the intensity of the ion with 220 m/Z. And as you can see, only in the sample where we spiked the acetaldehyde to the coffee, this compound is upregulated. And in the same experiment, but with the deuterated acetaldehyde, only the mass of 220 is upregulated in the sample where we added the deuterated acetaldehyde, which means that within our marker compound, there is one molecule of acetaldehyde incorporated. And then we performed some, basically some theoretical assumptions. And one of them was that our precursor molecule, as our marker molecule, has a neutral mass of 218 Dalton, and acetaldehyde has a mass of 44 Dalton. And one of the most common reactions is a condensation where a molecule of water is lost. And if we perform these theoretical implications, we end up at a mass of 192 Dalton for our precursor substance. And in the world of coffee chemistry, 192 is very significant for the quinic acid, which is the second most organic acid present in coffee. In order to verify this, then one of the possibilities would be to try to isolate these compounds from the coffee beverage and then do structure elucidation. But since this is really a minor compound formed, the extraction from coffee is not expedient. So we opted for a synthetic strategy where we synthesized these compounds. And besides, the compounds 1 to 6 were synthesized using the quinic acid as precursor. We also used chlorogenic acid as a precursor and the quinic acid lactone, since these are have the same structural features of the quinic acid and are also major constituents in the coffee brew. And in order to verify the structure, we performed then again high-resolution mass spectrometry experiments and NMR experiments. And just as a very quick example, the structure elucidation of compound 13 by means of NMR experiments looks like this. So this is an HMBC, an excerpt of an HMBC spectra, where the correlation or the couplings, the J2 and J3 couplings are shown. Very quickly, we can verify both structural features are present by the couplings of the protons 3 and 4, as well as the methyl group 2 with the carbon atom C1, and this confirms the formation of this acetal feature on the cis-diol function between C3 and C4. The second structural feature, which is the lactone, can be seen by the correlation between the compound between the protons on the diastereomeric CH2 groups, as well as the proton attached at carbon atom 5 with the carbon 7. Which confirms the structure. And in an analogous way, we elucidated the structure of these 18 compounds. And as I mentioned earlier, now a targeted approach was followed up in order to verify and to quantify the compounds, the reaction products in an actual coffee beverage. Therefore, we prepared several standard coffee beverages of different roast degrees, and then we added stable isotopically labeled standards. Here we used two compounds who were synthesized using deuterated acetaldehyde. They were added before the measurements. The samples were homogenized and then injected into a 6,500 Q-trap system. On the left-hand side, you can see the mass transitions. And here I summarized the concentrations of the compounds, showing you an overview of the different roast degrees of the coffees. And what we can clearly see is that there is a strong correlation between the concentration of our adducts and the roasting degree of the coffee, but also the concentration of the respective precursor. It plays a major role. So for example, the quinic acid reaction products show an increased concentration with harsher or with darker roasting. This can be alluded to the fact that more of chlorogenic acid is degraded into quinic acid, so the concentration of the precursor also increases with darker roasting, and that's why we can find more of the reaction products with darker roasted coffee. An inverse trend can be seen for chlorogenic acid. This can also be explained by the same trend, because the chlorogenic acid is known to be degraded during the roasting process, and the harsher the roast, the darker the roast, the less chlorogenic acid is present, and also the less of the aldehyde reaction products we can find. To come to an end almost, the next question that we always have in our working group is if these molecules have an intrinsic taste. So we performed bitter recognition threshold sensory experiments, and what we found is that all the precursor compounds do not have any bitterness to them, but the reaction products with chlorogenic acid, so the compounds 7 to 12, they do have a certain bitterness to them, and also the quinic acid lactone acetaldehyde adduct also showed a bitter taste. While the quinic acid adducts shown here, 1 to 7, they did not have any intrinsic taste. And just to give you a little bit of a comparison, these compounds lay in approximately the same range as literature known coffee bitter compounds. But the difference is that from these compounds, no direct impact on the taste can be expected from these reaction products because their actual concentration is way below their recognition threshold. So this means that not one of these compounds will have a direct impact, but there might always be some synergistic or additive effects. And of course, the aldehyde, the aroma that is bound, is no longer perceivable for the aroma of coffee. And with this, I am at the end of my presentation, and I would like to thank everybody for the kind attention. And if there are any questions, please feel free to ask.
Thank you very much, Michi, for the nice presentation.
You see my screen now? Right. So thanks for having me present our results here, especially because, as Christina just said, the main focus of our group is not actually mass spectrometry, but rather infrared spectroscopy. But we used proteomics to learn something important about blood serum of humans. And I hope that you will also find it interesting to learn about that together with us. Let me just see if I can switch the slides. Sorry. Right. So I would like to start with a plot that I think is one of the most shown, if not the very shown plot that I have seen in all the presentations that I have attended. And the reason for that is that this plot shows very nicely what's the main challenge in studying human plasma. It's the huge dynamic range, right? It ranges from albumin that presents almost half of all the proteins in blood, and all the way down to to the molecules present in tiny abundances. And ever since this plot has been published in 2002, or maybe even earlier than that, people have been interested in going lower and lower in abundances in order to find biomarkers. So biomarker, a molecule which changes a lot when a certain disease happens in the body. However, in our group, we take a different approach. We say, well, if it's a really serious disease, it's very probable that it will shake up many systems in the organism and change the abundances of many of these proteins in certain ways, in certain pattern. So what we do, we actually do infrared spectroscopy of all of these proteins at once and hope to determine certain pattern of changes. So we do that using infrared spectroscopy, as I just said, mid-infrared spectroscopy. So the wave numbers range here on the x-axis from, say, 1,000 to 3,000. And you can see this typical infrared spectrum of blood serum. There are many molecules that contribute to it. So it's mostly proteins, but then there is also lipids. There are also sugars. And all these spectra overlap one with another to provide this complicated signature. So what we did in our group, we collected blood of real people who have lung cancer. So this is the disease that we work with, it's lung cancer. And people who do not have lung cancer, but have related diseases. So they have difficulties breathing, or also non-symptomatic individuals, or healthy, simply put. And so we compared the infrared spectra of their blood, and then used some machine learning to see if there is any difference between this infrared spectrum. Okay. And we see, indeed, there is a difference. And this here is a so-called ROC curve. So this basically tells you how well we can distinguish the blood of people who do have lung cancer with the blood of people who do not. So the ROC curve, there are many ways you can read it, but what we use as a value out of it is the area under the curve. And for our case, it's 0.85, which might, so it can range from 50%, so 0.5 for a pure chance, being in one group or another, up to, of course, ideal case where it's 1, so 100%. And so 0.85 might not seem so much, but if you look at the other studies in the field, that's as good as you can get, more or less. So there are proteomic studies, purely proteomic, where they get pretty similar values out of 4 proteins or 7 proteins. Then there is also a large review that claims an AUC of 0.9, for example, using one particular marker, but this marker is not specific to lung cancer. It's a general inflammation marker. So you can see that with our 3-minute-long measurement, we are pretty much there with the specificity and sensitivity, but our technique does not tell us the identity of the molecules that we are looking at. We basically do not learn from our spectra which molecules are responsible for the signature. So, you know, Miki just presented you with 25 molecules that were identified. This is the smell of coffee. And we would like also to be able to say, okay, this is the flavor of lung cancer in blood. But nobody has done that before, or there were many attempts, but we were not able to tell which molecules are responsible for these features. So let me just explain what this graph is. So that's the difference between the spectra of lung cancer patients and the control group. And in gray, you can see the variability of the control group. So of course, people are different, so their infrared spectra are also all different. But you can see that there are areas where the spectra of lung cancer step out of this variability. And these are the areas that allow us to identify the patients. However, from this graph, we do not know which exactly proteins are responsible, as I just said, for this signature. And this is where proteomics comes into play, because that's exactly what it can do. We approached the group in Max-Planck-Institute of Biochemistry, the group of Professor Mann, who have in 2016 published their plasma proteomic profiling pipeline. So the beauty of that pipeline is that they can relatively quickly, scan through the samples. So in our case, it was almost 150 samples using just 1 microliter of plasma, although we of course delivered them, you know, slightly more than that. So they use a robot to automatize the whole process and do all the sample preparation steps basically in 1 vial. So what comes out of this process is a list of proteins present in each sample. And the first question that we had to address when we looked at that list, which proteins are important for us, which proteins should we take into account? And because we do linear spectroscopy, for us, the most important proteins are actually the most concentrated ones, the ones that are present in highest abundances. And then what is also important are the proteins where the concentration changes a lot between cancer and controls. So we compiled a list of 12 proteins that are important for us. And we measured their spectra. Here I'm showing 5 to give you an example. We measured their infrared absorption spectra of each of these proteins separately. And you can see that these infrared spectra are somewhat different, not too different, but still different enough that we are able to see the identity of every protein from this spectrum. Now, having their individual spectra, we also needed their so-called real concentration, by which I mean, you know, in mass spectrometry, you usually get values. So in our case, we had label-free quantification values, so LFQ values, which are very good for quantifying the relative change of concentration of every protein between samples. But they are not so good if you want to actually convert them into real concentrations, whether that's milligram per milliliter or whatever you want to call it. And the trick that worked for us was to actually scale them using literature values. So our control group was rather large. And for our purposes, what we did, we took the values, average values of these proteins for average healthy person and rescaled our LFQ values. And so that way we had very good quantification of the relative changes between different people, and also rather decent comparison between different proteins. What it was also important for us was the reproducibility of the proteomic profile. And here you can see the comparison of the coefficients of variation, so reproducibility of the measurement basically, for the technical replicas and for between person variability. And you can see that for all the proteins that we were interested in, the between person variability is by far higher than the technical replicas. And what this tells us is that when we see that there are differences in profiles of two people, we can safely assume that these differences are real differences between people and not the technical noise, of course, that is introduced in such a long pipeline. It's also interesting to note how high actually the coefficient of variation is for all the proteins. I was surprised to see that. Apparently people are really different between themselves. Our infrared spectra are also different, but on a different scale. You can see that the coefficients of variation for the whole profile are in general much smaller, but the trend is there that the technical noise is significantly smaller than the between person variation. So with all this pre-knowledge, now we were able to model our infrared spectra based on these 12 proteins that we identified. So we really took the concentrations, as this formula here suggests, multiplied by spectra, summed this up, and got our lung cancer signature. When we took the average lung cancer spectrum and subtracted the average control spectrum, you can see that we can reproduce most of the features here, the most important ones for sure. There is a couple of features that you might say, okay, they are not there in the model. So this one, for example, actually I have arrows showing that this ones and this one and this one. The reason for that is that these features are produced by lipids. That's the place where it's known that it's the lipids that absorb there. And so there is still space for lipidomics in this study, actually. So apparently there are differences between the lipid content of healthy people and lung cancer patients, and they might be identified in a further study. And this would, of course, improve then the way our comparison looks like. So we also compare the ROC curves, and with our 12 proteins, we can get almost to the same AOC value, 0.82 we get. So that's within arrow bar from what we measure in our infrared spectrum. So from the spectroscopic perspective, from the perspective of our group, the conclusion is very simple. It's the first time that an infrared signature of a disease in blood serum was explained thanks to proteomics, or thanks to any other actually technique. Because for sure, we are not the first group to see the differences between infrared signatures of different cancers, as well as Alzheimer, neurodegenerative other diseases. But usually there are only speculations about why there are differences. And we really took it to the point where we clearly see which proteins are responsible for these differences. And we are happy to share that this has been highlighted on a cover of Angewandte Chemie recently. Because it is an important step in the community. However, yeah, I wanted to show you that we now also suggest that any study that involves infrared spectroscopy would be combined with an omic profiling, so that one can identify the molecules responsible for the signal and then reconstruct infrared spectra and then clearly say which molecules are responsible for the differences in the disease. You might ask me, of course, if the omic profile can do that, then why would we do the infrared spectroscopy? And the answer is simple, it's quick and fast. It's a simple measurement, 3-4 minutes, and there is no sample preparation whatsoever. You take the fresh prepared plasma, put it in the commercial device, get the answer. That would be, I think, a very beautiful future we're aiming for. We are still not there though, because yeah, there is still a way to go to show that this is clinically applicable. However, since this seminar is about mass spectrometry, I would like to come to my conclusion regarding proteomics. And from my perspective, this study tells us that it's not worth ignoring classical plasma proteins just because there is a lot of them. And to illustrate that, I would like to show you that we identified 114 proteins in all samples. In general, there were many more, but they were not in all the samples present. And the 7 most important proteins in terms of differences between cancer and control provided AUC that was not higher within arrow bar than what we see in our simple infrared measurement. And the top most important proteins, actually the 2 most informative ones, were haptoglobin and alpha-1 acid glycoprotein, which combined provided already rather good AUC. And these are proteins that are very much present in human plasma, so among the 10 most abundant ones, actually. So they are important, and for example, combined, they are not used for lung cancer identification or screening or anything. There are papers only on haptoglobin separately and alpha-1 acid glycoprotein separately. However, it's important to note that everything we talked on these slides about is mostly related to inflammation. So the proteins that we identified is acute phase proteins, are acute phase proteins. And they are also changing in different inflammation processes. So we are not claiming that this signature is necessarily specific to lung cancer. However, we identify a pattern, right? So we see certain pattern of changes. This one is going up, that one is going down. And the question is how unique that pattern is to lung cancer, and or to different cancers, or to inflammation process in general. And this question is still to be answered, or we are in the process of answering it, actually. Of course, one can, in general, pose a question, what does it tell us about the biology of cancer? The big overriding question. With that, I would like to thank all the people who contributed to this study. So on the left, that's our group, although I'm not there, but you can see me on the screen. The bird team, as we call ourselves, is led by Dr. Gigl and Professor Ferenc Krausz. And this work wouldn't be possible without contribution from Matthias Mann group in MPI for biochemistry and his postdocs and PhD students, as well as the medical team who collected the samples, because that's where everything starts from, right? If you don't have the samples, then there is not much you can do. And the doctors do a lot to recruit people for our study. And on top of that, I would like to thank also this young member of my personal team, who was born exactly when the paper was in the process of being written. So if not his collaboration, this would never come true. And with that, of course, thank you for your attention.
Yeah, thank you very much for this excellent presentation on this very interesting topic. Are there any questions from the audience? I think you can either just raise your hand or unmute your microphone and ask away.
So I will have a question. But first, thanks for your talk. So Karen is in her maternity leave now, and we are also writing some paper together. So hope we can have the same progress. Okay, so at the end of your talk, I think you mentioned you can use some omics data, maybe proteomics or metabolomics data, to predict the spectroscopy spectrum. So do you know how accurate this prediction could be?
So in principle, we do see that with every added protein, we get better and better in prediction of every single spectrum of a single person. So, you know, I didn't focus too much on that, but the way we did the modeling was that we really modeled the spectrum of every person first as a first step. And then we averaged that and came to the lung cancer signature. And so when we add proteins, as we add them, we see that the modeling of every person's signature gets more and more precise. So we only did with this with proteomics. And okay, for this crowd really specialized, maybe I can mention that we also tried doing that with metabolomics. And the reason there that it didn't quite work was that we didn't find a proper trick to convert the data into real concentrations. So it's just didn't seem to work out. The numbers that we were getting for different people did not seem to match. And for example, the clinical data, because, you know, for some metabolites, we also have the clinical data like glucose or so. So one would have to overcome the challenge that mass spectrometry gives values, and these values need to be converted to some real numbers. But other than that, I think, yeah, this can get as precise as you like, as many proteins as you add, or as many molecules as you add, you can get more and more precise.